So this video is going to address how the MHC class 1 molecule will be loaded with peptides, so the antigen processing and presentation components. So we're going to start off with a CD8 positive T cell with its T cell receptor using the alpha and beta chain. And what we need to introduce about the MHC class 1 molecule is the fact that it's made of two proteins. One protein is called the alpha chain, and it's the protein that will load the peptide. And the other protein is a small protein called beta-2 microglobulin. So this is we call this the MHC class 1 molecule or the MHC class 1 complex, these two proteins together. So we use those terms interchangeably, complex or molecule. But they're found on the surface of most every cell in the body. What does this receptor interact with on a T cell? Well, we said that MHC class 1s interact only with CD8 positive T cells, not CD4 positive T cells. And you can see why here. The CD8 molecule on the surface of the T cell actually binds with the MHC class 1 alpha chain protein. So these two proteins interact. And the alpha chain and the beta chain of the T cell receptor also interact with the class 1 protein alpha chain. So anytime a CD8 positive T cell gets near an MHC class 1, this complex forms, and that T cell checks to see if the peptide loaded in there matches its antigen binding site. How does that peptide get in there? So you first have to remember where do these peptides come from? They come from the cytoplasm, right? Proteins present in the cytoplasm will end up being chopped up into pieces, small chains of amino acids called peptides, and loaded onto the alpha chain of the class 1 molecule. So what kind of proteins do you find in your cytoplasm? Well, right now, if you don't have any infections, they're your proteins. We're going to call them self proteins. And there's a self protein. So right now, you're presenting, if you don't have an infection, self proteins on an MHC class 1. And the T cell receptor, its antigen binding site, its variable region, has been trained not to interact with or identify strongly, bind strongly self-peptides. We'll see that when we talk about negative selection in the thymus in the next chapter. So it will check, the T cell receptor will check this peptide and say, oh, I don't bind it, I'm going to keep going. Now let's say you have an infection. Here's a virus infecting the cell. Right? Virus is in the cell. Virus makes proteins, so in order for the virus to replicate. So how does the cell process proteins in order to present them to T cells? Right? And see, that's what the virus wants. The virus wants to that's what the cell wants. The cell wants to take a bit of that protein from the virus and put that peptide on the surface of cells, and hopefully there will be one T cell in the body out of the billions of different T cells that have different T cell receptors. One of the T cell receptors will bind that peptide. So we have to talk about the proteasome, and hopefully you've learned about the proteasome in a class like biochemistry. The proteasome is a large protein machine in the cytoplasm whose job it is to recycle proteins. So in your cells right now, once your proteins live out their life, some proteins last 15 minutes, some proteins last days, but eventually proteins get used up, they get worn down. And so proteins go through the proteasome, and they're broken up into small chains of amino acids, and some of them are broken down even into single amino acids, so that you can take those amino acids, recycle them, use them to build new proteins. So the proteasome is a normal thing found in normal cells, used to break down proteins into small chains, and sometimes even single amino acids. So in a virally infected cell, the proteasome will also degrade some of the viral proteins. Not all of them, obviously, right? And obviously you don't degrade all your own proteins, but, there's some level of proteins being broken down by your proteasome. So you can make viral peptides using the proteasome too, and that's a good thing, because these peptides are going to be loaded onto the MHC class 1. As a side note here, you remember NK cell, natural killer cells from chapter 2? We said they secreted a cytokine called interferon alpha. Well, it turns out interferon alpha, that should be gamma. Let me cross that out. Gamma. Interferon gamma is a cytokine secreted by natural killer cells. So in an infected tissue, natural killer cells will go secrete the cytokine. And what the cytokine does is it actually makes the proteasome more likely to load peptides onto the MHC class 1. So we call this proteasome, this change in the proteasome. Now it looks like a slightly different proteasome. It's got a couple different subunits. We call it the immunoproteasome. So this proteasome is really likely to help load peptides on its surface. So, what else do we need to know about how these peptides are going to get into the plasma membrane, or going to get to the plasma membrane loaded on MHC I? So now we have to talk about the ER, the Endoplasmic Reticulum. Why do we have to talk about the ER? Well, anything you ship to the plasma membrane has to go through vesicles. Right? Vesicles start out in the ER. Membrane proteins all start out in the ER. So we got to actually get these peptides inside the lumen of the ER if we want to send them to the outside of the cell. How do we do that? In the ER, you have a protein complex called TAP. Stands for Transporter Associated with Antigen Processing. These two proteins are embedded in the membrane of the ER. There's TAP1 and TAP2, right? And they're responsible for taking peptides that are found floating in your cytoplasm, and those peptides go into the lumen of the ER, right? Self-peptides can go in there. Viral peptides can go in there. That's a great thing. So now we could send them out into the world. So this is the rough ER. You remember rough ER was started with ribosome. So mRNA can be translated here into protein. And so that's where we make our MHC I molecules. We make them in the rough ER. And so it's embedded into the plasma membrane. There it is. So now a process goes on called peptide loading. And your book goes into great detail into lots of different proteins that are involved here. You don't have to know them for this course, but your book talks about them if you want to know. But these peptides will end up getting loaded onto the MHC class 1 complex, specifically that alpha chain. There they go. So if you don't have an infection, you're going to load all self-peptides. If you have an infection, you will load both self and non-self peptides, right? I'm just showing one complex, but there are many. And so they all get loaded with peptide, right? And so if you're loading self-peptides, which you are right now, right, those are going to the plasma membrane. Right? If you have an infection, you're loading not just self, but non-self peptides. And those butt off into vesicles, and those also get sent to the plasma membrane. So this is antigen presentation. And so T cells can come by. Here I'm just showing one T cell, but in fact it could be any number of T cells. Will come by and check all these MHC class 1 molecules. And they will see, ooh, does my antigen binding site bind that peptide? We'll talk more about peptides in a later video. But if it doesn't, it'll check another one. It'll check all of them, right? And so all you need is one T cell receptor interacting with one of those peptides, and that T cell will become activated. And so that T cell will undergo what we call clonal selection, mitosis, and it will wander the cell, wander the body, killing any cell that's presenting that specific peptide. So that's antigen processing and presentation for MHC class 1. There will be a separate video for class 2. The last thing I want to say here is about TAP. There are some people born, and we talk about immunodeficiencies a lot, there's some people born with a defect in the TAP proteins, in the genes that make the TAP proteins. So you can look in your book and you can think about this right now. Imagine, what would happen if you didn't have the ability to make a functional TAP complex? What would be your immunodeficiency? What couldn't you do? And what would you be susceptible to? And so you can think about that now. You can find the answer in your textbook.